These here are the cells of a tomato under the microscope, and I'm going to show you now how you can prepare a tomato in such a way that you're also able to see those cells. Hi, hello, and welcome again. Microbehunter here. Well, a tomato, of course, is way too big to be directly observed under a microscope. Yeah, you have to prepare it somehow, and this is what I'm going to show you today. I sliced off a piece of the tomato. I washed the tomato first, of course, because actually I ate the rest of it. I do not want to waste any food. And I took a little slice, and then what I had to do is I had to scratch away the pulp of the tomato because I wanted to look at the thin tomato skin, because that skin is very thin and only approximately one cell layer thick, and that's what I wanted to observe, and because the cells there look kind of very nice and interesting. So I used the back side of the knife because the sharp side would have cut the tomato skin, something I don't want. So I carefully scratched it off. I used some scissors to carefully cut off this thin potato skin, and it did not work. I had such a big problems getting it flat on the microscope slide. It was really impossible. It always kept on rolling up. I tried to flip it on and around again, but there was no way of actually keeping it flat, so I had to restart all over again. And I decided to not cut it off right away, but kind of to leave it on the potato slice, and then try to carefully stretch it over the slide this way. And then I tried to cut it off, and I worked very carefully because it already started to roll in a little bit. But actually this proved to be a fairly effective way, because overall the potato skin did stay fairly flat. Of course, you always have to add a cover glass on top. This also keeps it more flat, and you have to add a little bit of water as well. Now I did not add any water right away, but I decided to add it after I put on the cover glass, and to allow capillary action to do the rest. So the water was pulled in beneath the cover glass, then the tomato was surrounded by water. This also makes sure that the cells do not dry out. And that's basically where we're able to get those beautiful cells here. So now let's have a look. Well, those cells, they look green. Now, of course, tomato itself is red. Why are they green? Well, this has to do something with the special optics that my microscope has. So the colors can be changed due to shifting of a filter. And these are the cells now in brightfield. You can see quite nicely the cell walls and the individual cells. And I was kind of happy with the result. Here in this technique, it looks a little bit better. You can see the cells a little bit better. I say I was kind of happy because the cells were indeed thin enough so that I could actually see the individual cells without any overlap. But here we do have some overlap. Here, this is the part of the tomato skin where it folded back onto itself, and this is actually at a very low magnification. But here you can see again, if we zoom in a little bit further, you can see now again the individual cell walls. I tried also staining it, but it did not quite work as expected. But it kind of shows that the preparation this way actually does work, and that's I would say also quite of a unique and a nice way of making plant cells visible. I just wanted to share that with you. Maybe you can also try it out. I just think it's always interesting to experiment around. Don't give up too quickly if things don't work out. And then maybe you're also going to be successful. For me, that's all I would like to share with you today. I wish you all the best. Happy microbe hunting as always, and see you in the next video. Bye-bye.